welcome to another A-level biology video with Mr. Phillips. Today my camera doesn't seem to be working, so we're just going to have sound, but it will all be good, and we'll go through the content. So we're going to look at the kidneys, and we're going to look at how the kidneys do something called ultrafiltration and selective reabsorption today. So first of all, you need to know where the kidneys are found. So effectively, the picture shows you there where they're found, on your back, or the back half of your body, above your hips, I suppose, and just below your rib cage. Now, kidneys' functions are they excrete excess water and salt, okay, and that affects osmosis, obviously. It also affects the blood pressure as a result in your body, so it's important to control. They excrete ions such as potassium, sodium, chloride, and hydrogen. So if we have excess ions from our diet, this is how we get rid of them. But also they make sure too much don't go as well, because we do need them for our healthy living and our nerves and things like that. It's regulation of acid-base balance, so enzyme activity is not affected, so some acids and some bases will come out that way, and obviously hydrogen ions and sodium and hydroxide ions will affect that. And it's the removal of nitrogenous waste formed from the breakdown of amino acids and nucleic acids. So we're not going to look at it today, but the liver is what produces urea. Okay, so the liver produces the compound called urea, and that is made for something called deamination. It's from the excess amino acids in your diet. And also, the kidneys are involved in production of something called a erythropoietin, which is a hormone involved in the production of red blood cells. We're not going to look at that particularly today. So what do you need to know about the kidneys and sort of their blood flow? Well, it's really important, obviously, the blood flow, because that is how we get things to the kidneys. And the kidneys are in what we call the urinary system, and the kidneys get the blood via the renal artery. So the blood enters the kidneys via the renal artery, and it comes in, it goes into some capillaries, which we'll talk about later in a few moments, the afferent and efferent arterioles, and then the blood leaves via the renal vein, which is then taking away the cleaned blood effectively. And obviously there's other tubes involved, though we do need to know, it's worth talking about now. Once the urine has been made, the urine leaves via the ureter, which is going down to the bladder, and then the ureter, so then from the bladder to the outside world, it's the urethra. Okay, so ureter and urethra. And obviously, urine is stored in the bladder once you've made the urine. So in year 13, you are very likely to do a kidney dissection and a diagram of the kidney. So just to, I think, point out a few things here. There's a tough capsule on the outside of the kidney, which is basically dark because it's got such a good blood supply. That's where the cortex is next to it. And then we've got the medulla, which is light region. Now we'll talk about this. This links to this structure called the nephron here. So each individual kidney bit, each individual working part of the kidney is called a nephron, and it covers both layers of the cortex and the medulla. Try and always remember this, because you need to know in terms of like the medulla oblongata and cortex. Cortex is always in the brain, that is. Cortex is always like an outer layer, and medulla is always an inner layer. Now, if you were asked to draw this, you might have to be given something like this and say, right, draw a scientific diagram, just to really remind you quickly. If you asked to draw this for a required practical, you'd expect to take up the space given. You'd need a sharp pencil and use continuous clear lines, no shading. Demonstrate appropriate proportional sides. It must be fully labelled, and the lines must be drawn with a sharp pencil and not crossing over. There should be no shading or colouring in a diagram. So what we're looking at then, we're talking about the kidneys, we're looking at individual tubes. So there's millions of tiny tubes called nephrons, which is a functional unit of the kidney where the filtration and reabsorption of the blood occurs. And it all starts in somewhere called the Bowman's capsule, which we'll be talking about today. Then it goes to the proximal convoluted tubule, then to loop of Henle, then to distal convoluted tubule, and then finally the collecting duct. So we're going to look at the Bowman's capsule and the proximal convoluted tubule today. And that is where we sort of start our journey. So if you just look at this diagram here, the blood comes in via something called the afferent arteriole, and it goes around a series of capillaries here. And then those capillaries is under very high pressure before it leaves, okay, and the high pressure forces some components of the blood into the nephron of the kidney, and that is the Bowman's capsule that you can see there. So you might see a diagram like this, much more modern sort of picture. Exactly the same thing you can see there. So the blood coming in from what we call the afferent arteriole, the name you need to learn, and it leaves by the efferent arteriole. And as it's in this Bowman's capsule area, the glomerular capillary, which is the blood, so the blood's called the glomerular capillary, the kidney part's called the Bowman's capsule. That is where the filtration occurs. So if we have a look at this, this says explain using your knowledge of ultrafiltration and difference in concentrations of the table above. So if you look at concentrations in the blood, so that would be in your glomerulus capillary, and then concentration in the filtrate. So this is in the actual kidney here. You can see water, you end up with the same concentration, so all the water goes across. Amino acids, you end up with a very small concentration, but the same concentration. Glucose, the same. Urea, the same, and so on and so on. But the one thing that stands out is protein. And that is because very few, if any, proteins get filtered, and that is due to the size matter. Okay, so molecules with a size greater than 69,000 relative atomic mass cannot fit through the filtrate. So the ultrafiltration we're about to talk about literally is based on size, like you would think filtering is. So here we go. So ultrafiltration at Bowman's capsule. So here you can see quite a few bits, and they're quite important diagrams to sort of go through with you. So if we start off here, the blood comes in via the afferent arteriole, as I've said. Okay. Now, if you notice here, the width of the afferent arteriole, okay, and then if you look at the width of the efferent arteriole that the blood leaves. Now, what that shows you is that the blood coming in, there's more blood coming in than can leave by the efferent arteriole. Okay, so this leaving here. Now that means that there's sort of like a queue effect, like a traffic effect almost, that more blood's getting in than can get out. So that causes the pressure in this glomerulus to be high, which starts to force the molecules into the Bowman's capsule of the kidney. So it's high pressure. How is high pressure maintained? Because the afferent arteriole is higher than the efferent arteriole. Now you need to understand then, well, how is it filtered? What is it? Just little holes, or what is it? So we're going to have a look at that. So we just need to know the blood supply is very good. It's supplied by a branch of the renal artery called the afferent arteriole. That splits into a load of capillaries, and then that is called the glomerulus. And the kidney part is the Bowman's capsule. And the capillaries rejoin to form the thinner or narrower lumened efferent arteriole. Now, when you get to the Bowman's capsule, then you've got three actual layers. And you saw that on the diagram that we just saw a minute ago. There's three layers. The first layer is the endothelium of the blood capillary. So the blood capillary itself has tiny little pores in it. So we're talking about this, the wall of this blood capillary here has got tiny little holes in it, which would allow only small molecules to go through, so tiny pores. We then have a basement membrane. So between the epithelial of the capillary and the endothelial of the Bowman's capsule, there's actually another layer of molecules, okay? And this is made of glycoprotein and collagen fibers, and basically it's a mesh-like structure, okay, which makes it very, very strong because it's collagen fibers, which will withstand high pressures, okay? But because it's a mesh-like structure, only small molecules are going to be able to fit through that mesh-like structure. So it's a filter, effectively. And then finally, you get this next layer, which is called the Bowman's capsule epithelial cells, and they're called podocytes. Okay, and basically these have got finger-like projections, which again, if you look at the blue diagram on the previous slide, you can see, but they don't fit tightly together. So they've got little gaps between them, allowing the molecules to go through. And you would be expected to be able to describe that in detail. So you'd be expected to say the first layer is endothelium cell of the capillary with lots of pores, basement membrane made up of glycoproteins and collagen fibers in a mesh-like structure which fills it, and podocytes in the Bowman's capsule epithelial cells, which have gaps between them, allowing further filtration. And that will allow then the smaller molecules to get, but not the bigger molecules. So effectively, there are holes in the cells, are larger than most cells, but large molecules like proteins can't fit through. So it stops the molecules due to size, and this is your key number here, okay, which is the relative atomic mass or relative molecular mass should really be of 69,000 or greater cannot get through, and most proteins have a relative atomic mass of 69,000 or greater. red and white blood cells cannot get through as a result, okay, but the smaller molecules can get through, and that is why we then lose some things. So what things can get through? Well, you've got urea, obviously, that's the key thing we're trying to get rid of. Charged molecules like sodium and chlorine and those ions, okay, water can get through, and glucose can get through. Very few amino acids can get through as well. But glucose is a problem because we don't want to lose any glucose in our bloodstream. It's our energy supply, so we have to selectively reabsorb any of the glucose, all the glucose, and we need to selectively reabsorb any water or mineral ions that have been lost that we actually need. The glomerulus filtrate rate, so the amount of filtration that happens, is 125 centimeters cubed every minute. So it's very quick and very efficient, but that doesn't all become urine because of selective reabsorption. So you just need to be aware of this pressure thing again, just to remind you. So you can see there the pressure that we have is because the afferent arteriole has a wider lumen, okay, than the efferent arteriole, and you need to know those two terms. So the lumen of the afferent arteriole is wider than the efferent arteriole, and that means more blood can enter the glomerulus, can leave at a higher rate, and you get sort of a traffic jam effect, which maintains a high pressure. And it causes also the water potential to be higher in the blood than the renal capsule. So you always go from a higher water potential to a lower water potential, and that means that the majority of the net movement is into the Bowman. Okay, so once we've done the filtration, we then enter the proximal convoluted tubule, the next part of the nephron. And this is a bit where basically the fluid in the tubule is basically identical to the blood. Apart from the fact it's got no blood cells and no proteins in it, it's got all the things that we need: water, glucose, things like that. If it was to pass out of the urine, we would die, okay, so that you wouldn't survive. So we need to use selective reabsorption, okay, and that happens along the proximal convoluted tubule. Mainly, 85% of it happens, and then after the proximal convoluted tubule, it's much more just about water. So this diagram is more complicated than you need, okay, but I think it's quite useful for us to look at. So basically, fluid tubule endothelial cells have microvilli on them, which increases the surface area of the cells, okay, so you should know that. That hopefully makes sense to you because we've done that lots already. And what happens is sodium is pumped in using facilitated diffusion, and co-transporter proteins therefore also take glucose and amino acids. So we just have a look here, and I'm going to start basically here, like we would have done with number one, like you would have done with protein and glucose digestion in the intestines. So what happens is there's active transport happening here. So it's a sodium potassium pump, which we still haven't done massive amounts on yet, but we'll just accept it for the minute. Sodium is actively pumped out into using ATP into the bloodstream. That causes a lower sodium rate, or a lower sodium concentration, I should say, in the cell of the epithelial cells of the nephron. That means then that sodium wants to move in from the proximal convoluted tubule into the epithelial cell, but it can only do it if it goes through a co-transporter protein and it brings in glucose and amino acids. So in your cell then, what will happen is the sodium will go from being low concentration again to being a bit higher concentration because you've just moved it in via facilitated diffusion. You'll also have glucose, which I'm just going to represent by a letter G, and also we've got amino acids. Now what that does is that then brings all these solutes in here, that then lowers the water potential, okay, so you get a lower water potential. If you remember, the symbol for water potential is like a W with a stick underneath it, okay. That then causes water to follow via osmosis from a higher water potential that's left in the blood, in the proximal convoluted tubule, because you've got lots of water left in there, okay, to the lower water potential now in the epithelial cells because you've moved all the sodium and all of the glucose out. So some of the sodium, all of the glucose. And then what will happen is, again, the sodium will be pumped through actively and the glucose will naturally diffuse through into the bloodstream. So in terms of what then happens and what gets reabsorbed, well, mainly passive osmosis. The water is the main thing that's happening. The concentration of solutes in the filtrate stays just about the same because you lower the volume. There is only then when it leaves about 45 centimeters per minute leaving the proximal convoluted tube to the next part of the nephron, which we'll look at next lesson, called the loop of Henle. So that just dropped from 125 that has entered. So it shows you the amount of reabsorption that happens. Okay, and then urea, there will be some urea diffusion back and forth, but it's not a lot. Okay, but the point is, it's obviously your blood's constantly going past your kidney, and this is constantly happening. Only 50% of the urea, because of diffusion, therefore stays